They carry the functions.
Mr. Mac?
They function the functions. Yeah.
I'm so sorry, but can you light this part up?
Can we put a light on?
Yeah.
Yeah, I gotta put the ones across the middle. Is that alright?
Yeah.
Okay.
Whoa.
Okay.
Thank you.
Is that alright for everybody? Still see okay?
No, but
So yeah, the proteins, remember we called them little tiny, what? Little tiny machines, yeah. So these are the proteins.
Today we review.
근데 유닛 7은 딱 쓰고 가야 돼. 유닛 7은 쓰고 가야
Right.
돼.
So these guys are all about helping things happen. Proteins. We need proteins to make things. We need proteins to do things. Right? Proteins are what's making everything happen inside of the cell. Right? And we know, right, the cell is sort of that smallest unit of life. Right? Everything that goes back to living stuff, eventually we can sort of zoom in on it and we can work down to the level of the cell. And proteins are what's in there making all the good stuff happen. Okay. So that's the general idea. How are they formed? What's their structure? Remember, structure always relates to function in biology. Structure always relates to function. So what's the structure of a protein? Okay, that's where we're going to go to, but what about if we sort of start with the building blocks?
Amino acids.
We start out with amino acids, and what's the structure of those guys?
Linear.
So we've got amino acids, and we have a structure. What's our structure? Yeah, that chain. We got a chain on the bottom and a chain on the top. What do we call those guys? The base chain and the side chain. Yeah, our base chain looked something like this. Right, and that was the same. We have how many different types of amino acids that exist sort of in life? We've got 20 different types of amino acids, 20 different amino acids going on. And every single one of them has the same base chain, right? All 20 of them have the same base chain. So we'll throw that guy on the side. Whoa, that didn't work very well. There we go. Base chain. And then you said something about a side chain, and where does that fit in?
On top of carbon.
On top of the carbon, yeah, right. And we know we've been talking about bonding and stuff like that. We can see, remember, nitrogen has got 3 sexy singles around the outside, so it makes 3 bonds, right? Carbon we know has 4. We can see over here 1, 2, 3, 4. This guy needs an extra one. So we've got our side chain, which is sticking off the top. And what do we usually use to represent that? We usually throw in an arm. So we set our base chain the same in all 20. And our side chain different. Come on, and there we go. Right, and that's what makes proteins sort of unique, right? We can make all these different types of chains by linking these amino acids together. So if we look at one amino acid, what is it that we would call that? What would be a general term that we can use across the board for all of our different macromolecules? A general term for one unit, for a single unit.
Monomer.
A monomer, yeah, we can call it a monomer. So our amino acid is essentially a monomer, right, of the larger polymer, which we call the protein itself. Right? So by linking these subunits together, by linking each of these pieces together, we can create a much larger chain of amino acids. And that's what we saw. What was it? We were just studying something and we saw linking, we were linking amino acids together. What was that we were talking about? That's what we call it once we've linked them all together, but what was the process we were just studying?
Hydrogen bond.
No, sorry, not in one of the review sessions, actually in class. Protein synthesis, right? And translation, right? Transcription was the one where we were building the mRNA strand, and then we took the mRNA strand out to the ribosome, and then we translated it, right, from nucleic acid language into protein language. And that's where we were linking together all of those amino acids, right? The tRNA would deliver an amino acid, and then the ribosome would shift over, so that tRNA was in the P site, and a new tRNA would bring a new amino acid into the A site, and they would link up, right? That was the process that we were talking about. That is how these proteins get built. That's how we link together these amino acids to make these giant protein chains, right? So in order to stick these guys together, what's the process? How is it that we stick these together? We looked at how we stick glucose molecules together the other day to form right, different polysaccharides, starch or glycogen or something like that. How do we stick these guys together?
Nitrogen.
Thing, yeah, we got to be a little more specific.
Water molecule.
Which is it, an H bond or an H2O bond?
H bond.
An H2O, yeah, we're gonna remove water, right? But it's not actually gonna be H2O that's involved in the bond. What's it gonna end up sticking together? Be careful. You just guess it. You just wait. You have to think before you say anything. Yeah.
Oxygen from that carbon is going to connect with the hydrogen. Yeah, hydrogen from the other amino acids, and then the C and the N.
Yeah. So we've got this side from one is binding with this side from the other, right? So if we put a couple of those guys down there together and see what's going on. And then we have another one going over here. Put our side chains up there. Right? Then you said a 2, what's going on? Yeah, these guys here are going to bond, right? So we're going to break that bond there. We're going to break this bond here. And these guys are going to come together, forming H2O. What happens to the H2O? Yeah, it just disappears, right? It just becomes part of the cell. It just goes in there. There's always water floating around all over the place, right? Water is used, you guys will study this for more if you go into like IB biology or AP biology. You need to talk a little bit more about enzymes, which are specifically how proteins really become helpful in the body, right? But forever, water is either being stuck in between different things in order to break them apart, right? Or it's being pulled out of things in order to attach them together. So water is constantly being used in both directions in terms of the reaction. So we get those guys pulling out, and then what happens? What's left over? Yeah, we're going to end up, right, this over here, this sexy single, right, these sexy singles have been broken apart, right? That guy's been given back to the oxygen. This one's been given back to the hydrogen, and that's why they end up holding hands and going off as a nice couple over here, right? And carbon's still sitting there with one sexy single left over, and so is nitrogen on this side, and that's where that last bond gets formed, right? Between the sexy single there and the sexy single there. And those guys form a bond. So that we have the bond going from carbon to nitrogen. Right, so if you look at that sort of on the side, what we end up with is this. Right, and that would kind of be the zoomed out version of what's left here. We'd see the bond to the carbon, up to the double bond oxygen, right, and then we see that bond going over to the nitrogen, up to the hydrogen on the top, and then off to the rest of the molecule on that one. Yeah, how are we doing with that? Thoughts, questions, things that you're looking at going, huh? We're okay? Yeah? All right, so once we take a whole bunch of these amino acids and stick them together, you all right? You with this one? Good, yeah? Be slapped in the face. Not by me, of course. Yeah, you can take your choice. I'm sure there might be a couple of them that'd be okay that would helping out. So anyways, once we take these amino acids, right, and start sticking them together, once we start joining them together, what is it that we form? Our polypeptide chain, right? Our amino acid chain. Why do we call it a polypeptide? Morning.
Poly is many.
Poly means many.
Because the bond is called a peptide bond.
Yeah, this bond right here, that green bond, is called a peptide bond. So when we make a whole bunch of these guys join together, then we have a polypeptide because we have a whole bunch of peptide bonds, right, that are linking all these amino acids together. So we form this long chain, right, which is essentially what we were doing in translation of protein synthesis, right? We form this big long chain of amino acids, and then structure leads to function. So keep going. How does this relate to our structure, or how does this relate to our function? Okay, why does the hydrogen bonding form? What's causing that to happen? Say again? The polarity of what? Of the different side chains. Yeah, so those different side chains. So depending on the sequence, depending on the order of the amino acids, right? Those amino acids will start folding and pulling together and twisting, right? Some of them will be slightly positively charged. Some of them will be fully positively charged, right? Some of them will be negatively charged. And those will start pulling together on each other. Sometimes you'll have positives and positives near each other. And what do? Yeah, they're going to repel each other and push each other apart so that those parts aren't going to twist together, right? They're actually going to make sure that they push apart from each other, right? And what ends up happening is you get this three-dimensional structure that starts forming, right? And when that three-dimensional structure forms, now that's where we start giving the protein its function, right? Its shape then gives it its function. And we haven't really talked about how its shape gives its function other than the fact that it does, right? And again, if you keep studying it further up, you'll see how different shapes will help with different functions, right? Certain shapes can help break different things up. Other shapes can help pull things together and make them one, right? We looked at ATP synthase as a protein, as an enzyme, right? ATP synthase, remember what that was sticking together? What did it stick together? Way back, first semester. It took ADP and P, right? ATP synthase just took ADP and P, and it brings them close enough together that they stick together, right? And then ATP is formed, right? Even if you look at a ribosome, right? A ribosome is essentially a protein, right? It's just a couple of proteins that are pulled together. And what's its job? What does the ribosome have? How does its shape help its function? What's its function, first of all? The ribosome. To make proteins. And how does it make proteins? Reads the mRNA, so it reads the codons in the mRNA, and what's it bringing in? Bringing in the tRNAs that have the amino acids. And the whole structure of the ribosome is such that the mRNA slides in on the bottom, right? And there's two spaces for the tRNA molecules to come and sit nice and close right beside each other. And when those tRNAs are nice and close right beside each other, what does that enable? What does that allow to happen? Say again? Between what? The two amino acids, yeah, up on the top, right? If the ribosome was set up somewhere different, I mean, so the ribosome that we looked at, right, had a structure that was kind of like this, right? And it had the two sites that those guys came into, so we had an mRNA flying through here, we had a tRNA popping into each of these sections here, right? And those amino acids were held nice and close, okay? Which allowed them to bind with each other, okay? What if your ribosome was set up so that it looked kind of like this? What if this guy went like that, and your A site went that way, and your P site went that way? Yeah, now when the tRNAs pop in, right, now you've got amino acid over there, and you've got amino acid over there, and they're too far away. They can't join together, right? So simple changes in structure all of a sudden completely shift the functional capabilities of the protein, right? It's because they bring those tRNAs nice and close together now that those amino acids are nice and close, right? And that's what enables them to form. Yeah, and from there, right, once we've got our folding, twisting, we've got our shape, we've got our function, the protein goes off into the cell wherever it needs to be to do whatever it does. Thoughts, questions? What do people think? Anything that we're missing from that? Kelly B? Nothing? You guys okay? And that's what I'm asking you guys. Is that all? Is that all? Is there anything that we can...
Mr. Mac,
Yeah.
do we need to know homo and hetero?
Oh, homo and heteropolymers. What are those?
Homo is same thing, hetero is simply more like the same thing.
So give me an example of how this would be associated with a protein. How would you have a homopolymer for a protein?
Yeah.
Same what?
Side chain.
Same sequence of what?
Side chain.
Yeah, the same sequence. Yeah, so the same sequence of side chains just means the same amino acids, right? The side chain indicates the amino acid, right? So if you've got, you know, an example, lysine was one of the amino acids we were looking at, right? There's lysine. That would be a homopolymer. It's just the same repeating unit over and over and over and over again, right? But a heteropolymer would just be made up of different pieces, right? Different units. So you'd have lysine, you'd have valine, you'd have arginine, you'd have methionine, you'd have whatever. Isoleucine, you just keep going to be all different amino acids chunked together. Anything else? That makes for a great multiple choice question.
Oh.
So hetero is just different.
Yeah, hetero means different, homo means same. We just paused to see if anybody had any questions. I was just waiting to see if anybody would speak up. No?
No.
What are we learning?
We just learned about proteins. Now we're going to move on to nucleic acids. If we're good. We're good?
Yeah.
We don't have a whole lot of time, so I don't want to just sit there and wait for everybody the whole time. Okay, so we're moving along. We need a new chart. So going to nucleic acids. What's going on with these guys? Why do we care about these guys? Why are they important to life? DNA is an example of a nucleic acid. It's not the reason why they're important. I guess you could argue that. Yeah, it stores and carries genetic information. Right? It stores and carries genetic information. Right, and two examples of those.
DNA, RNA.
DNA and RNA. Right. What do we know about DNA?
Double helix.
It's got a double helix structure. Okay. What is a helix? It's like a spiral, right? Yeah, it's like that coiled shape. Imagine yourself on that spiral staircase, right? So that's a helix shape, right? So we've got a double helix, which means we have two of them actually spinning around. I'm going to give you one of those metal ones back. So DNA, we've got the double helix. What else do we know about it?
Codes for RNA.
Say again?
Codes for
It
RNA.
codes for the RNA. Anything else?
A, T, G, C.
A, T, and C and G, right, are the what?
For nitrogenous
They're
base.
the nitrogenous bases, yeah, of the nucleotides, right? A, T, C, and G are the four nitrogenous bases that DNA uses. And A always binds with T, right? And C always binds with G. What's it stand for? You remember DNA? Not dioxy. Dioxy means two oxygen.
Deoxy.
Deoxy, yeah. Deoxy means what? What we've been talking about deoxy stuff now when we're talking about the respiratory system. What's deoxy mean?
No
Without
oxygen.
right? Without oxygen. So not without actually, yeah, low oxygen. Because deoxygenated blood doesn't have zero oxygen in it. It's just got very low oxygen. Right? And those guys, the ribose, which is the sugar inside of DNA, doesn't actually have no oxygen on it, just missing one oxygen, as opposed to regular ribose. So it's called deoxyribose. deoxyribonucleic acid. And then this guy down here, RNA, I'm just going to move this down slightly for us so we can see a little bit easier. Go away. It's what do you say, single what?
Single helix.
Single helix, okay. RNA, yeah, I got to get rid of this table. Single helix. What else?
Codes
for protein.
Codes for proteins now, right? Codes for that amino acid chain. And
ACGC.
A binds with what? U or me. Yeah, and C binds with G. A binds with U and C binds with G. So it's exactly the same, right? Except that T is replaced with U. T is replaced with U. So some of the basic ideas there. If we start looking at these guys, how does our structure look? How does our structure compare? You want to do the brown. You don't want to do the brown. Anybody want to do the brown? It's kind of like a Mario Brothers sound or something like that. So DNA, right, is going to have the brown and the brown, and it's going to go around.
Can you write the differences?
What?
Can you write the differences?
How would you even spell that? I don't even know where to start. You guys know what you call those things?
WRAO.
What do you call that? When you make, like, spell the sound. Yeah, good.
Oh my god.
Nice. So, yeah, DNA, double helix structure, right? What do we know about each of these strands?
What?
Is that for the student council stuff?
Yeah.
How did it go yesterday? I wasn't here.
Good.
Did it go well? Did it go well? Cool. You were doing a thing. Was anybody else doing a thing, a speech? Tori was also doing a speech. How'd they go? Well? Oh, yeah, the boys did really well. We finished 4th and 5th. Girls finished 2nd. Yeah, girls did very well. We finished 4th and 5th. Those are 3 schools. What? Figure that one out. Anyways, let's jump back here. We only got about a minute to go here. What's up with our structure here? What's up with the brown structure? It's what? It's a double helix. We see that. Anti-parallel, right? So this is an anti-parallel structure, anti-parallel strands. If we have one of these strands being a polymer, right, because it's nucleotides all joined together, and we have another one of these strands all being a polymer, nucleotides joined together, we have 2 polymers interacting with each other. What do we call that one?
Mono.
Remember that was the 3rd term. There was heteropolymer, homopolymer, and starting with C.
Copolymer.
Copolymer. There it was. The anti-parallel strands. This was a copolymer. Right? And if we have these anti-parallel strands... Come on, give me some order here. Give me some direction. Yeah, one of these is going to be 5' to 3', and the other one, therefore, is going to go in the other direction.
Mr. Mac?
Yes?
So heteropolymer and copolymer are not the same thing?
No, a copolymer just means more than one polymer interacting with each other. So if you have 2 polymers joined together, right, like we do, we've got hydrogen bonds holding all these guys together in the middle, right? So that's 2 polymers that are then interacting with each other. Anything else we need to throw up here quickly? You guys? Yeah, so the structure of a nucleotide, right? If we look at a nucleotide specifically, right? The nucleotide specifically, we've got a phosphate that's then attached to a sugar, which is then attached to a nitrogenous base, right? And if we have a look at that guy inside of here, we can see our different, these would be our nucleotides. And over here, they'd be going the other direction. Yeah. We okay?
Yeah.
Any other questions?
No,
That's all. Man. We took like 3 days to cover this.